Good morning, guys. Welcome to another Biology with Miss Cherry. Today we are looking at lesson 3.6. We're going to be looking at plants, we're going to be looking at how they minimise water loss, but also how they exchange gases. So to start with, have a go at these 3 questions, pause and play again when you're ready. So hopefully you've had a go at these 3 questions. Photosynthesis is a way of plants turning sunlight into energy. Okay, and this is a biochemical reaction. It's the inverse of respiration, aerobic respiration. So we have got 6 CO2 plus 6 H2O plus the sunlight plus the chlorophyll gives us C6H12O6 and 6O2. Notice that when we say we're turning the sunlight into energy, it's not actually making ATP in the same way that respiration does, but that glucose can then go on to respire. And we can break that down. Now, for plants to exchange gases, I'm hopeful that you will know some kind of plant anatomy, but also maybe not, we don't know. Plant exchange gases through their stomata, okay, those pores on the underside of the leaf. And these are part of the leaves being specialised for gas exchange, but we're going to have a look at some other structures over the next few slides that will hopefully tell us a little bit more detail. Well, they will tell you a little bit more detail. Yeah, they will. Okay, let's get started. So as we've said, this is lesson 3.6. We're looking at gas exchange in plants. We've done the insects, we've done the fish. We're now going to go into plants, and then we're going to go into humans for a little bit. And we're going to look at how they limit water loss. Because we've got to remember that plants are very, very susceptible to environmental change because they can't move. So let's get started right at the beginning, and we're going to look at gas exchange in plants to start with. Now, we know that plants require oxygen for respiration. Okay, that means that they've got to somehow get oxygen in. But they're also going to require carbon dioxide for photosynthesis. So that's got to come in too. What we can do with plants, which makes them slightly different to other biological organisms, is that gases produced from, say, respiration, that carbon dioxide, can then go on to be used for photosynthesis. But have a think, what does that mean for the rate of gas exchange with the external air? Is it going to increase? Is it going to decrease? And why? Now, at any time, the gas exchange with external air is reduced because remember, we can use gases for other processes. Use gases for other processes. And what this means is that sometimes plants don't need to take in as much gas as they would do at another time. Okay, so remember, when we're using these square brackets, they mean concentration. So the concentration of gases exchanged at any one time is variable. It can change. It can increase or decrease. And I want you to spend a couple of seconds, maybe just thinking about it. What kind of things are going to affect the concentration of gases and the rate at which they're exchanged? Think about what kind of things are going to affect the way that gases leave and enter a plant. Now, ultimately, the concentration of gas exchange is going to depend on the rate of photosynthesis and respiration. Okay, and this is again controlled by other environmental factors that you should know from GCSE, but might not be able to remember. And if you can't remember, then maybe take a look back at bite size and things like that to help remind you. So have a go at this for me. You're taking a look at how the light and the dark affects the rate of photosynthesis and gas exchange in plants. Okay, so you're thinking about what happens in the light. Maybe you're looking at carbon dioxide, maybe you're looking at oxygen, and then what happens in the dark and why. So pause, play again when you're ready. It should take you a couple of minutes. I would suggest this is maybe a time to work with other people, maybe message them, maybe put it in the chat box, ask some questions, and then play again when you're ready to go through the answers. So your answers should have hopefully looked something like this. In the light, some carbon dioxide for photosynthesis comes from respiring cells, but most of it comes from the external air. Remember, photosynthesis is the process of plants producing that glucose that they need to respire, to lay down things like fruits and stuff like that. Okay? So any extra carbon dioxide that's needed that isn't produced from those respiring cells diffuses in from the outside air. Okay, notice that we're using our key words. We're relaying Unit 2. So we're revisiting, we're talking about the movement of gases and things like that. Oxygen, produced in photosynthesis, mostly diffuses out of the plant. Okay, we're going to have a look at how that happens in a second, but most of it is going to leave. Now in the dark, photosynthesis isn't going to occur. Okay, the plant is entirely reliant on diffusion to get oxygen into the leaf. Entirely reliant on diffusion. If we've got no photosynthesis, because we haven't got any sunlight, we're not going to have that light energy to power the reaction. We're going to find out next year as well about how that actually happens, but it's not going to work. Photosynthesis is dead, not happening, until the sun comes up again and photosynthesis can start. So now we've had a look at the rate of photosynthesis and how that varies between the light and the dark. We're actually going to take a look at the gas exchange process itself. So have a think and use the knowledge again that you got from GCSE, because notice we're just building up why it's useful that plant leaves are thin and why it's also useful that diffusion takes place in air as opposed to in a liquid. So just spend some time thinking about it. Obviously we'd be having a discussion in lesson, but we can't do that right now, can we? I mean, you can talk to me if you like, but you probably won't. So the first one, looking at why it's useful that the leaves are thin. It means that no living cell is particularly far away from the air. So there's a really short diffusion pathway, and this means that the diffusion process is going to happen quicker. It means that our gases can be exchanged more readily and at a moment's notice rather than spending forever and ever getting through all the leaf. The second one, diffusion taking place in the air as opposed to in a liquid, and definitely more so than in a solid, means again that it is quicker. If you remember, gases have much more kinetic energy. They're going to be able to move more readily. And because of that, we're going to get a much faster equilibrium reached than in liquids or in solids. Now in plants, there's not really a gas exchange system. It's very, very passive. Okay. Conversely, if you think back to when we looked at the fish, they had that countercurrent flow in their gills, and in insects they had the spiracles and the tracheoles and all of the contraction of the body and things like that. In plants, they just got some holes. So it's very, very passive. It doesn't rely on energy. So how are they adapted? You can take a look at the photo, or you can just carry on playing, because we all know that's what you're going to do anyway. How are the leaves adapted for this gas exchange? So to begin with, we've got the GCSE favourite that everybody loves to talk about, the stomata. Now, before we move on, I want to make really, really highly clear, I want you to flag it up in your notes, the stomata are the pores, they're the holes. Okay, they're not the bits around the outside, those are the guard cells, we're going to look at those in a second. But the stomata, the pores, means that gases can come into and out of the leaf really quickly, really easily. And because of these stomata, when we start looking at in the next slide at all of the air spaces, it means that no cell is particularly far from that external air, which is only going to emphasise that short diffusion pathway and make the movement of gases a lot more smooth, easy and effective. Now, as I said on the previous slide, we are looking at these air spaces, or the spaces in the spongy mesophyll layer where I'm putting all of the shading, and those are effectively like air chambers. Okay, that mesophyll or middle layer has a lot of air spaces that are all interconnected. So even if one stomata is damaged or one stomata is open, we're going to get a lot of transport of gases into all of those air spaces really quickly. Again, more efficient, quicker, better for the plant. And the final adaptation is to recognise that the mesophyll has a large surface area. So all of these air spaces mean that we've got a lot of space, a lot of surface for gases to diffuse into. If you imagine this spongy mesophyll layer was quite flat or not even spaced out at all, you wouldn't get as much surface area. It's that lumpy curving and infolding that is beneficial. Bigger the surface area, the quicker the gas exchange. The more efficient it is, the more the plant is going to be able to photosynthesize, it's going to be able to respire, it's going to be able to grow. We spoke about the stomata previously. They are the pores that are mostly found on the underside of the leaf. That's not to say that they're found at the top. They can be in some cases, but they're mostly found on the underside of the leaf for various reasons that we will look at when we do the transpiration lesson later on in module 3. Now the stomata is the pore. As you can see, it's that gap, that hole, looks very similar to the spiracles of an insect. And each stomata is controlled by 2 guard cells. They guard the pore. They open and close it. If your name's not down, you're not coming in. Let you in and out, or let gases in and out of the leaf. You're not going into and out of a leaf. Now the guard cells are able to open and close the stomatal pore depending on the concentration of water available in the plant. They control the opening and closing of the stomata via osmotic pressure, via osmosis and the inflow and outflow of water from those guard cells. Before we move on though, I want you to think about why the gas exchange in plants, so the opening and closing of the stomata, is a double-edged sword. I.e., why is it good, but also why is it bad? Let's pause and play again when you're ready. So the good things about the stomata, the guard cells, is that plants need to exchange gases. We need to get oxygen in, we need to get carbon dioxide in, we need to release waste gases. And the stomata allow that to happen. However, at the same time, when we look at the transpiration lesson, this is going to be more relevant. Opening and closing the stomatal pore could result in dehydration of the plant, water loss. If you imagine it like breathing with your mouth closed, your mouth stays quite wet, quite spitty for want of a better word. Whereas if you start breathing with your mouth open to exchange those gases, your mouth starts to become quite dry quite quickly. And that's because of evaporation processes that are similar to what happens in the plant. Now we're going to take a look at the adaptations of guard cells. We're going to go through these in turn. We're going to have a look at how each of these adaptations is beneficial to prevent excessive water loss. Because remember, that's what we're looking at today. We're looking at gas exchange and then minimising water loss in plants and other organisms. So the first adaptation is that our guard cells have got a thick inner wall that you can see on the diagram by the slightly darker green line on the inside, and a thin outer wall. When we've got the guard cells opening, so when they're turgid, the pore is wide. Now if that doesn't really make sense, we'll have a go at a diagram. So I've got 2 guard cells shaped like little beans, because that's what they are, but they are equal width in the walls all the way around. If water moves into those cells, both of them, then in becoming turgid, all they're going to do is swell to become fat little sausages. Okay, that's not going to help open the pore. Conversely, I'm going to go over to the left-hand side now and use a nice orange colour. If I've got slightly thicker walls on the inside, which I will draw here and here, if water moves into those cells, then those thicker inner walls are going to provide some resistance. Okay, so they're just going to stretch a little bit, and that gives us a nice stomatal opening, whereas the rest of the cell is going to curve around. So you can see with that thicker inner wall, that provides a better hole, a better pore, and a wider stomata for gas exchange to happen. Okay, it's going to enable maximal gas exchange, which is ultimately only going to serve to benefit the plant. The second adaptation is related to the third in the way that these guard cells respond to water levels in the plant. So guard cells will only swell when water is in excess in the plant. In excess means that they've got enough to survive and a bit extra. Okay, they've got surplus water. Oh, extra. When this happens, water moves in to the vacuole, into that plant cell, into the guard cell, and it's going to open the stomatal pore by making those guard cells turgid. Okay, they're swollen. A bit like if you fill your cheeks up now with air, so you keep your mouth tightly closed and you fill your cheeks up with air and then push your cheek, that's turgid. Okay. Now, the reason why they only swell when water is in excess is because they've got water that they are able to lose. Okay, so water can exit the plant, so we can start transpiration happening without it negatively affecting the plant, and at the same time, we can get gases coming in and out. And it's going to help balance the water content of the plant. It's going to stop it getting too turgid. It's going to stop it getting too inflexible. And the final adaptation, as we said, linked to the second adaptation, is that guard cells deflate or become flaccid when the water is limited. So when they don't have water that they are able to lose, the flaccidity or the movement of water out of the guard cells means that the stomatal pore is closed. Means that gas exchange can't happen, but also is a protective mechanism, because water ends up being contained inside the plant. It can't get out of that stomatal pore. You're not going to lose it. Your plants aren't going to start drooping. Alongside the stomata, most plants also have the following two adaptations. See if you can name them. Number 1 and number 2. What are they? What are they there for? How do they help reduce water loss in a general plant? So the first one shows the waxy cuticle. It's found on the upper and lower surfaces of the leaf. And what it is a waterproof layer that stops water being able to leave through the leaf surface through transpiration. So it means that water loss can only occur out of those stomatal pores, which is where our second adaptation is. Those guard cells are supporting the stomata. They open and close depending on the water concentrations of the leaf. They help to control water loss and minimise it if the plant is in a series or a situation of stress. Other plants that live in different conditions will have other adaptations. And we're going to spend the rest of the lesson looking at something called xerophytes. Okay, so I'll write how you say it. Xerophytes. And these are plants that have got adaptations to allow growth and reproduction in dry conditions. Okay, so in hot climates, if you think about things like that, there's not going to be a lot of water in the soil, in the sand, and the water is also going to evaporate rapidly. So even if you've got that thick waxy cuticle, the stomata are never going to be open, so you're not going to be able to do gas exchange. So instead, these xerophytes have got specific adaptations that mean that they can survive properly and well in these dry conditions. So a couple of examples of xerophytes that you might have seen are conifers, so coniferous trees, tall, leafy ones, and marram grass. And this is that itchy, quite scratchy grass that you find on sand dunes a lot. So if you go down somewhere like Camber Sands down on the south coast, there's lots of that on the dunes before you actually get to the beachy, watery bit. Now, before we take a look at those adaptations, think about this question. Are plants that grow in very cold and icy conditions likely to be considered xerophytes? Why or why not? So think about it, write your answer down, and then play again when you're ready to go through it. These plants are considered to be xerophytes. The water is locked into the ice structure. It's not freely available. It can't be absorbed into the plant. It can't move via osmosis because it's frozen. So actually, these are, while icy conditions, considered to be dry. because that water cannot move freely. Now for the next few slides, we're going to take a look at the adaptations of these xerophytes. You might want to do this in a table, you can do it in notes. There is quite a lot of information coming though, so it's up to you how you do it. I just need to see evidence that you have been making these bullet points, these notes, and adding some knowledge to your noggins. So the first adaptation for water conservation, that's what we're looking at, the ability of these plants to hold on to water, is the fact that we have got a thick waxy cuticle, so thicker than normal, and sunken stomata. So that means that they are slightly deeper away from the surface. And I want you to think about why these are good for the plants. So we've got a few reasons as to why these sunken stomata and the waxy cuticle being thick are quite useful. So the first thing is that the thicker the waxy cuticle, the less water loss we're going to get via osmosis from the surface of that leaf. And this is generally found in evergreen plants, things like holly, that if you touch the leaves of them, they feel ever so slightly more shiny than other leaves. Now, that's because of their thicker waxy cuticle. It means that they can survive both hot and cold conditions really effectively. And the final adaptation is possibly the most key and the most relevant in terms of exam answers. And this is the sunken stomata. So I'm just going to scroll across a little bit and put that there. So if you imagine the surface of the leaf, I'm going to draw it in green, like this. So we're looking at a cross section, so as if we've cut a leaf in half and we're looking straight down the middle of it. The stomata being sunk means that they are further away from the surface than they would normally be. Okay, so we've got the thick waxy cuticle here, and then those sunken stomata. Now what that means is that in that area where the stomata is sunken, that air is still. Okay, we say that because that air is still, we've got a stagnant microclimate. And in doing this, it means that any water that is lost is going to give us a humid area of air. It's going to be quite damp, wet air. Now, if we've got a lot of water in that little microclimate right by the stomatal pore, then water isn't going to have a significant concentration gradient to move out of via osmosis. Okay, so normally I'm going to draw a couple of diagrams that hopefully might make it a little bit simpler. Standard stomata, so this is on the surface of the leaf, stomata. You might have in the spongy mesophyll, you might have a high concentration of water that you want to get rid of, and in the air outside the stomata, you might have a quite low concentration of water, and you've got that steep concentration gradient. So that means that water is lost easily. However, with that sunken stomata, we might have a concentration of water still quite high in the spongy mesophyll, but because we've got that microclimate, because the air can't move around that sunken stomata, it means that there's a slightly higher concentration of water in that area. Okay, so the gradient is a lot shallower. And in doing that, it means that water is lost less easily. Okay, the diffusion gradient isn't as steep, the water loss doesn't happen as quickly or as excessively as as when the stomata aren't sunken. Okay, so this is only in those xerophytes, the dry conditions. Please remember that. Our xerophytes also have a reduced number of stomata. Now, I know you can't see the labelling on these diagrams particularly well, but at the top we have got a cactus, and at the bottom we have got a rainforest plant where the air is quite humid and it rains a lot, hence rainforest. So again, spend some time, think about why this is useful. Why does it help that the cacti have a lot less stomata than a rainforest plant? Now, the first thing we need to say is the way that we actually explain this in an exam situation. So we have to say that the density of stomata, or the stomatal density in the xerophytic plants, or the xerophytes, is lower. Now, what this means is that we prevent the amount of space available for water loss. Yeah, we don't have as much holes for water to move out of. They're not going to move as much water out. Common sense. But this is where that double-edged sword comes in, because we also have a reduced capacity for gas exchange. Okay, so because we've got those fewer pores, means that oxygen and carbon dioxide can't come in and out, means that maybe these plants are going to grow slightly slower. So remember, in the exam, they're not just going to tell you, oh, they've got a reduced number of stomata, so it means they can't grow as effectively. They might say, why does a reduced stomatal density mean that these xerophytic plants grow slower than a normal rainforest plant? Use your noggins. Stretch your brains. We've got another adaptation here. Here at the top we have got, I think it's a fir tree, like a Christmas tree kind of thing. It's coniferous. And at the bottom we have got a cactus. Now, what do you notice about each of those plants? Think about what I've zoomed in on. What is the key features of those two kinds of plants? Now, one of the key things about xerophytes is that they don't generally have big leaves. They don't have those big palm tree style leaves. They've actually normally got either needles or spines. So, we say needles on things like pine trees, and then we've got spines or spikes on things like cacti. And if we've got a reduced volume of leaves or a reduced number of leaves, then we're going to have a smaller surface area for them. And the smaller the surface area, the fewer stomata we've got, so the reduced water loss we have as well. There's no holes for water to get out. Remember, there's no holes for water to get out. There's no holes for gases to exchange either. Important. Now, with these spines and needles in the cacti and things like the pine trees, they're actually quite rounded, almost circular, and we're going to have a look at these rolled leaves and shapes in a second. But because they're quite round, the surface area to volume ratio is really, really small. So if you imagine if you're making I don't know if any of you have seen any of those videos online of making like an ice cream cone. They start with a huge sheet of wafer. And then by the time they've wrapped it up into the cone shape, it's much, much smaller than it was originally. And it's the same with these spines or these needles. So if they've got a massive surface area, by the time they've curled themselves up into that spine shape, they're going to have a lot less surface area exposed to the environment. And again, smaller surface area, reducing that water loss. Now, I don't know how many of you have got cacti at home, but some of them are spiky, some of them are a little bit furry, some of them are spiky and furry, and some of them actually look quite fluffy. And this is another one of those adaptations. So before we talk about how plants have little hairy leaves, think about why we have hair on our arms. What is it there for? You should have done it at GCSE looking at thermoregulation. What is that hair doing? So pause and then play again in a second once you've had time to think about it. And think how that relates to plant adaptations. So in plants, that hairy needleness or the fluffiness again helps get that microclimate. So remember we said in the sunken stomata region that it traps the humid air in that little pit. Likewise, if we've got the surface of a leaf like this, and it's got lots of little hairs like this, then air is going to be trapped in that layer. It's the hairs are almost providing like a little barrier for air movement. So that means that air is now nice and humid. And again, that humid trapped air means that we get a reduced concentration gradient. And in some cases, these xerophytic plants will double up on these adaptations. So if we take a look at the bottom picture, we've got a photo of a TEM image, so transmission electron microscopy image, and we can tell it's TEM because it's 2D, it's flat. But we can see that we have got this marram grass with both the sunken stomata, okay, so sunken stomata there, but also the hairy leaves. So they've doubled up on that microclimate just to maximise the water retention in those plants. And the final adaptation, again, we've got a cross-sectional image of some marram grass here. You can see already that we've got the sunken stomata and we've got the hairy leaves, is the curling of the leaves as well. So these curled leaves are where we take a flat leaf, much like when I said about the cone, and they curl them up nice and tightly, means that we're going to get a really, really small surface area, as we spoke about with the needles, but also means that the stomata are confined into that inner infolding. So inner layer has the stomata. They're not exposed to the air. And that's helpful because it means that we're going to reduce the airflow past those stomata. We're going to reduce the rate of transpiration or water loss from the plant. And also in conjunction with those sunken stomata and the hairy leaves, we keep that microclimate to reduce the rate of water loss. And sometimes we can get plants that have all of these adaptations, and likewise sometimes we can get plants that only have one or two. It all depends on the situation, and it's really important therefore that you read the stem of the exam questions. All of them. Look at the diagrams, look at the photos, look at everything that you're given to help identify which adaptations the xerophytes from your question have. So I've done a lot of talking, it's now time for you to have a go. Complete the How Science Works. Not only desert plants have problems obtaining water, it's around lesson 3.6. I don't know what page it is. You guys can find it, use your noggins. It'll be around the limiting water loss in plants chapter. So pause, and then on the next few slides we'll go through the answers. I'm not going to talk through them because I don't have the questions with me, but I'm sure you can cope with knowing that question 1, 2, 3 are there. Okay, so pause. Should take you probably about 10-15 minutes. Doesn't matter if it takes longer. We're chill. We're doing our best now. Pause and play again when you're ready. Alright, answers wise. Question 1 is here. So rain rapidly drains through the sand for 1 mark. Dunes are normally in windy places, so reduce the water potential in a 2nd mark, and thereby reducing the water potential gradient, so there's quicker water loss for the 3rd mark. Each one of those points that's got square brackets with a 1 next to it is a mark. If you didn't get any of them right, correct your answers. For question 2, the soil is very salty, 1 mark, with a low water potential for the 2nd mark, so root hairs have difficulty absorbing water by osmosis for the 3rd mark. And finally for question 3, in winter, the water in the soil is frozen for 1 mark and cannot be controlled by osmosis for the 2nd mark. Remember, those icy plants are still xerophytic, they're still xerophytes, they still can't access that water, it's locked up. For question 4, this was a big one. So there's lots of points that you could have had, or lots of marks within these 4 points that you can see. First point, photosynthesis is enzyme controlled, 1 mark, and is therefore regulated by temperature, 2nd mark. Remember, if you think back to topic 1 when we did proteins, temperature has a significant effect on enzyme activity. In cold conditions, enzymes work slowly, so limit the rate of photosynthesis. That's just 1 standalone mark. There is a reduced need for light as photosynthesis occurs slowly, for another mark. And water climates have a higher rate of photosynthesis and large leaves with a big surface area to capture more light energy. That's your final mark. And if you didn't get all 5 marks, take a look at the question. Think about the information that you got given, maybe in the paragraph surrounding the questions, but also things that you know already. If you didn't understand any of these marks, please ask. Tell me if you're not sure. Ask me to explain them, that's absolutely fine. I'm here if you need me. Now this is a little exam hint and is really, really key because they like to bring in modules together. So this topic links really heavily with module 2 when we looked at osmosis. When explaining adaptations of xerophytes, always make sure you're talking about the water potential gradient and the resulting effect on osmosis and diffusion, and not just about the water movement. Okay, so don't just say like, oh, the water concentration is higher here than here, so water won't move. No. If you've got a curled leaf, you've got a microclimate of humid air, which means that the water potential gradient is lessened or lowered, and in turn reduces the rate of osmosis and the movement of water out of the plant. So you notice how we've turned from a very bland, basic, frankly quite rubbish explanation as to the water movement and changed it around to be the most detailed A-level answer that we can. Make sure you're using your keywords. This is why you've still got those key terms and we haven't given up with them yet. If you're not too sure about all of the keywords, go back to topic 2, have a look over osmosis and water potential again. If you're confident, carry on with your keywords for topic 3, add that detail. Now it's your turn to have a go at explaining how do the following adaptations prevent insects from losing too much water. So you can see here I've got a little body of a caterpillar. We've got the small, quite chunky surface area to volume ratio, a waterproof shell made of chitin or a waterproof exoskeleton, and finally the spiracles. And this is your chance to do some research, your chance to do some summarising. Why is each of those a good adaptation to stop insects losing too much water? Now we're going to take a look at some exam questions. So we've got about 4 marks of exam questions and then we'll go through the answer. This is from June 2016, so quite a while ago now. A student investigated the distribution of stomata on leaves from 2 species of plant. She removed small pieces from the lower surface of the leaves of each plant species, and she then mounted these pieces on separate microscope slides. She then counted the number of stomata in several parts of the epidermis on each piece of leaf tissue using an optical microscope. Suggest appropriate units the student should use to compare the distribution of stomata on the leaves. So this is 1 mark. What units could they use to compare the distribution of stomata on the leaves? Pause and play again when you're ready. Second question. The pieces of leaf tissue examined were very thin. Explain why this is important. So think back to the stem that you were given. It told you the fact that she's put them on a slide and she's using an optical microscope to look at them. Why is it important that the leaf tissue is thin? Give 2 reasons why it was important that the student counted the number of stomata in several parts of each leaf tissue, not just in one. 2 reasons why it was important that the student counted the number of stomata in several parts of each piece of leaf tissue. 2 marks. Pause and play again when you're ready. And the final question. One of the 2 plant species used by the student in this investigation was a xerophyte. Other than the distribution of stomata, suggest and explain 2 xerophytic features the leaves of this plant might have. Suggest and explain 2 xerophytic features that the leaves of the plant might have. This is 2 marks again. Pause, and then when you play again, we're going through the mark scheme. So the first question was looking at the distribution. Okay, so distribution is not the same as abundance. Distribution is to do with the abundance in an area. And you have to be able to give what you're looking for, so the abundance of the thing, so you could have said number of stomata, and then the area that you're looking at, so per millimetre squared or per centimetre squared. But notice in the mark scheme it says reject per micrometre squared. We're not looking that small. Okay. Now for question 2, give reasons why it was important that the leaf tissue was thin. Remember, an optical microscope is the same as a light microscope, and this is one of those standard questions that they love to ask and repeatedly ask as well, just in different guises. So it was thin to ensure that there was a single layer of cells, or to prevent overlapping of cells, or to prevent too many cells overlapping. And that's the first mark. And the second mark is to say so that the light can pass through them. Okay, so remember, we need to get that light through so we can visualise our image. Question 3, why is it important that we sample in a few different areas? Maybe that the distribution may not be uniform in all of the areas, and so it is a representative sample. Okay, maybe that you should have said or could have said in another way that there may be more or fewer stomata in different areas. It does say ignore any mention of anomalies or randomness or bias. So maybe if you're looking down a microscope, you might be biased towards the areas that look like they've got more stomata. it's a bit of a meh point for this question. And the second mark for question 3 is to ensure that you obtain a reliable mean. And for this question, it does say that you can use average in the answer, but most situations at A level will accept mean, but not average. So just try and use the word mean from here on in. And the final question is the adaptations of the xerophytes question. And I'm going to leave this on the slide, but notice that for each of them, it was the suggest and explain. So I'm going to underline the suggests. And then notice how after all of them, there is so. You need to have both the suggest and explain for each point, the correct explain as well, to ensure that you get those marks. Okay, so if you just said hairs and sunken stomata, that's no marks because you haven't explained them. Okay, so I'm going to leave this here. I'm not going to read through all of them. I hope you found that lesson useful. I know it was a bit of a long one, I'm sorry. And the next time I see you, we'll be talking about humans. Yes! Alright, see you later, guys. Have a lovely day. Bye-bye.